Welcome back, everyone, to what promises to be an unusual fall semester, but at least we're starting it off with a really interesting talk. It is my great pleasure to introduce a longtime friend and colleague, Professor Stephanie Yano. Professor Yano received her Bachelor of Science from McGill in 1996 and completed her PhD in cell biology from University College London in 2001 under the supervision of Nobel Prize winner Paul Nurse. Since 2015, Dr. Yano has been a professor in global health within the School of Public Health and is cross-appointed to the Department of Medical Microbiology and Immunology at the University of Alberta. We've heard from Professor Yano a few times over the years about her work in malaria, particularly malaria and pregnancy, and I am very excited to hear about the latest advances in coming up with a vaccine against this devastating parasite. So welcome, Professor Yano, and I think we're ready for your talk.
Thank you so much, Joel, and thank you very much for inviting me to speak. I was actually thinking about it that the last time I gave Global Health Rounds was the first week of March in 2020, which seems crazy to me now how long ago that was and just how much has changed. So anyway, what I'm going to talk about today is, well, it's not COVID. We're going to talk about malaria. And really a lot of malaria research has been on the back burner for the last 18 months. But I want to start this by going back further in time, really, hold on, let me put that, to discuss a little bit about the history of the development of a malaria vaccine in general. And then I will transition this into some of our recent work on trying to develop a vaccine against malaria in pregnancy. So if we go back to the 1950s, this is after World War II, there was a really concerted global campaign to try to eradicate malaria. Now, I should preface this by saying that this was mostly in developed countries, so in Europe, in North America, Australia, and so on, and actually focus on developing countries that were primarily endemic for malaria was, okay, hold on, there we go. Was actually not a priority. But regardless, after World War II, there was a lot of focus on trying to do mass drug administration with chloroquine and also treatment with insecticides to try to get rid of mosquitoes. And there was this, illusion that malaria elimination was highly achievable. So to give you an example of this, Paul Russell wrote a book which is ironically titled Man's Mastery of Malaria. It was published in 1955, and he's quoted as saying, "While keeping in mind the realities, one can nevertheless be confident that malaria is well on its way towards oblivion. Already as a malariologist, I feel premonitory twinges of lonesomeness, and in my own organization, I am now a sort of last survivor." Now, we'll contrast this with the reality of malaria today. So this is an infographic recognizing World Malaria Day in 2020 and highlighting some of the latest data from 2019, which gives you this stark reality that actually half the world's population, so about 3.2 billion people, are currently living in areas where they're at risk of contracting malaria. In 2019 alone, there were 229 million cases of malaria. There were 409,000 deaths, and more than 2/3 of those deaths occurred in young kids under the age of 5, primarily living in sub-Saharan Africa. So clearly we have failed in trying to control malaria. Now, why? So one of the issues I think is really first and foremost in answering that question is the complexity of the biology of the parasite. So just showing you this life cycle here where you can see what happens in the mosquito host and what happens in the human host, I think you can already tell that it's an incredibly complicated life cycle, excuse me, which illustrates how well adapted this parasite has evolved to these two different hosts. So I want to briefly take you through the life cycle. If we start up here, I hope you can see my cursor. If not, somebody can please let me know. But we have a mosquito, which is carrying the sporozoid form of the parasites in its salivary glands. And then that female Anopheles mosquito is taking a blood meal from a human in order to feed her eggs. As she does this, the sporozoids are injected through the skin and go into the human host. Now, the first stage here of the sporozoids going through the skin, they then quickly make their way into the liver. They spend about 7 to 10 days in the liver. And although the person has no idea they're infected, they're completely asymptomatic. Regardless of that, the parasite, meanwhile, is undergoing massive replication, producing tens of thousands of new parasites and differentiating from a sporozoid form to a merozoid form, which is these little circular forms here. So when these merozoids emerge from the liver, they're now adapted to infect the red blood cells, and that leads to the erythrocytic cycle. So what happens in this cycle is the merozoid invades the red cell. It digests the hemoglobin as a source of food and amino acids. It undergoes replication, producing about 16 to 20 merozoids per cell, and then finally ruptures the red blood cell membrane, releasing these merozoids, and the cycle starts all over again. This cycle, the asexual cycle, typically lasts about 48 hours. And this is the stage where people experience the symptoms of malaria, the fever, the chills, and so on. And this is also where you get the major pathogenesis associated with malaria, where you can die from malaria. However, it's also the point where you can diagnose it and treat it if it's recognized very quickly. So aside from the asexual cycle, you also have a very small proportion of or subpopulation of these parasites that somehow receive a signal to evolve into the sexual form. So the male and female parasites, which are called gametocytes. So if that person is then bitten by another Anopheles mosquito, and she takes up these male and female parasites in her blood meal, these parasites will then undergo fertilization in the midgut of the mosquito. They will undergo meiosis and produce these sporozoids that then make their way up to the salivary glands, and then we have the whole cycle that can continue. So when we think about why haven't we eliminated malaria since the 1950s, I believe that the complexity of the life cycle of the parasite plays a major role, and I'll be touching on this when we talk about vaccines as well. But we've also discovered that many of the interventions that we've tried to introduce to deal with malaria have been countered by the parasite selecting, or evolution selecting, for parasites that have evolved certain adaptations that can counter those interventions. So for example, when chloroquine was widely distributed globally back in the 50s, we found that about 10 years later, the parasites had evolved specific mutations in genes that could actually pump out the chloroquine from the parasite itself. So that's led to drug resistance to chloroquine. Now, just about every parasite strain in the world of falciparum malaria is resistant to chloroquine. And we've seen this with other subsequent drugs that have been brought in to try to treat malaria. Even the mosquito has evolved to counter our public health interventions. So we've seen insecticide resistance in the mosquito population, so resistance to DDT and so on, that we've been using to try to get rid of the mosquito vector. Another very interesting adaptation that's more recent, and as far as I know doesn't have a name, so I kind of arbitrarily called it diagnostics evasion. And what I'm referring to there is that it's the parasites evolving resistance to rapid diagnostic tests, or RDTs. So these are tests that were brought in over the last 20 years or so, maybe 10 to 20 years. And what they do is they detect a specific antigen or a parasite protein that's in the blood. And what we're finding in many parts of sub-Saharan Africa and South America, that the parasites have evolved a deletion of the gene that encodes that protein that's detected by these diagnostics. So it's really quite amazing to me that you can have this selection for these parasites that can even avoid a diagnostic that could lead to treatment and wipe out a parasite population. And the last one that I'll be talking about a little bit more today is immune evasion. And malaria has evolved a number of different immune evasion mechanisms that, again, speak to the ability of this parasite to to really establish itself in us, the human host, and also in the mosquito. But ultimately, if we can develop a vaccine for malaria, then we don't need to worry about all this, back and forth between us and the parasite, and we could really break that cycle and eliminate this pathogen from our lives. So this is another cartoon of the life cycle, but really looking at it from through a vaccine lens. So here you have your sporozoite stage, the liver stage, the bloodstream, and then you have the sexual differentiation in the mosquito. And it's just pointing out the different types of vaccines that are in development to try to break this cycle. So we have pre-erythrocytic vaccines, so these are targeting the liver stages. We have blood stage vaccines. These are generally trying to prevent invasion of the red blood cell, of the parasite into the red blood cell. And then we also have transmission blocking vaccines that are targeting specific proteins expressed by the gametocytes in order to prevent that stage in the mosquito that would then allow ongoing transmission. Now, these vaccines have been in development for decades. This table here is just showing you the number of clinical trials for malaria vaccines since 2000. So this is looking at vaccines that are registered on clinicaltrials.gov. We have vaccines for the two major species of Plasmodium. So Plasmodium falciparum is the primary focus of vaccines. This is the most pathogenic or virulent Plasmodium species. It's the one associated with most of the deaths from malaria. But Vivax is an equally prevalent species. It actually probably has a broader distribution globally than falciparum, although it is thought to be perhaps a bit more benign. So if we look at the number of, these are just different types of vaccines, and then if we look over these 5-year periods, we can see that there was a real boom in vaccine clinical trials, particularly from 2006 to 2010, but then it kind of plateaus. I mean, we don't have a huge increase in the number of trials going forward from there. And if we look at the total number of trials completed over this 20-year period, we have 145 trials. Now, that may seem like a lot, but I will show you the data that I pulled up for COVID. And if this was a live talk, I would love to get people in the audience shouting out to me how many vaccine trials you think have gone ahead for COVID, but I can't really figure out how I would do this on Zoom. So I'll give you the short answer, and that's 910. So this is over just the last 18 months. There are 910 vaccine trials for COVID-19 that have been registered on clinicaltrials.gov. So one thing that I do think is very important is that making a vaccine against a virus like COVID is not as complicated as making a vaccine against malaria. And this is highlighted in this quote from a review article where they're talking about how tricky it is to make a malaria vaccine. And they say parasites have much more going on than viruses, making this much harder. Their multiple life stages have been explored, mostly without success. And this quote, which I particularly like, I've highlighted it here, is that malaria vaccine development has been a graveyard for really great ideas. So it's not for lack of trying. There's been a lot of creative ideas about trying to make a malaria vaccine, but the reality is we've learned about a lot of stuff that doesn't work. So what I'd like to do is share with you some of the some examples of the most current malaria vaccines that are either already licensed or in late stage clinical trials, and give you a feel for what are some of the challenges in developing a malaria vaccine. And then for the last part of my talk, I'll share with you some of the specific challenges in developing a malaria vaccine against pregnancy-associated malaria, and talk a little bit about our own approach to that. So the two vaccines that I'd like to highlight for trying to prevent a malaria infection in all populations, but particularly in kids, both target this liver stage of infection, this early stage when the sporozoites are coming in to infect the hepatocytes. And the first vaccine that I want to talk about hasn't been licensed yet, but it's really at very advanced stages of clinical trials. And this is the vaccine that's produced by a company in the States called Malaria, and it's called the PFSPZ for sporozoid vaccine. And what's interesting about this vaccine is it's a whole parasite vaccine. So the idea is to vaccinate someone with the sporozoites to stimulate the immune system to recognize many different antigens expressed by the parasite at that stage. And just to give you an idea of how you make this vaccine, so what they do is they have an insectary. They feed the mosquitoes on blood that's infected with Plasmodium falciparum. They have the parasites, the sporozoites will be in the salivary glands of these mosquitoes. They dissect out the sporozoites from the salivary glands. And then what they want to do is attenuate these sporozoites. So they don't want them to be fully alive because they'd be infectious and that would be very dangerous. But they also don't want them to be completely dead because then that limits the amount of antigens, excuse me, that can stimulate an immune response. So what they do is they use radiation to attenuate the sporozoids. So they're mostly dead, but not completely dead. And then they have to cryopreserve this vaccine in order to preserve it, and that means keeping it at very, very low temperatures. So if we look at some of the advantages of this vaccine, so first of all, it targets the liver stage. So that's that first stage when someone is infected by the parasites. So if you could have an effective vaccine that blocks that first step, you could prevent even the stage where symptoms appear. So you really could block all of those downstream effects. And by using a whole parasite vaccine, you're presenting the immune system with many different antigens. So there should theoretically be less immune pressure for the parasite to avoid the immunity. And also there's a likelihood that some of those antigens at least may be conserved across different strains of Plasmodium falciparum, which, as I'll talk about a little later, is another major challenge in malaria vaccine development, is that these antigens that are expressed on the surface of the parasite tend to be highly polymorphic. And the other advantage with this vaccine is that it can be combined with other vaccine strategies, which I won't talk about today, but there's some interesting combinations of this radiated sporozoites with like a chemoprophylaxis with chloroquine, for example. And I'm happy to talk about that at the end if anyone's interested. But of course, there are a number of challenges with this vaccine. First of all, the manufacturing is very difficult to scale. I know Malaria, they used to do all of the dissections of the sporozoid or the salivary glands manually, but now I believe that's all automated with robots. You need to be able to really closely control the amount of radiation that you give to these parasites. You don't want to have breakthrough infections. Cryopreservation makes it challenging. It creates all kinds of cold chain issues, particularly if you're trying to deliver that vaccine in remote communities in sub-Saharan Africa. So these can lead to concerns about reproducibility of the vaccine. Another potential issue is that the vaccine is given by IV. And I know a lot of clinicians have concerns whether that's feasible to give a vaccine, particularly for young kids. And really having the healthcare workers that have that knowledge on how to deliver a vaccine by IV injection. The other reality is that the vaccine efficacy for PFSPZ isn't fantastic. So these are some more recent data where you have looking at roughly 6 months post-vaccination in different populations and also different ways of challenging people with malaria. So the best data that they've reported from Malaria is that when they immunized US volunteers that had never traveled, never traveled to a malaria endemic country, never had malaria, and then they challenge them with what's called a controlled infection. So they grow the parasites in the lab and then they give them a very specified dose of a particular strain of Plasmodium falciparum. They found that there was 65% vaccine efficacy. And for a malaria vaccine, that's not too bad. I mean, with COVID, we're looking at 90–95%, but for malaria, 65 in the grand scheme of things is pretty good. But when they started doing trials in Africa, they started in Mali, so in West Africa, and here they vaccinated people, and then people were just exposed naturally to infection. So because it's a highly endemic area. And here they found that the vaccine efficacy had decreased to 52%. What was interesting though is that when they then went to Tanzania in East Africa, and this time they're actually given a controlled infection as the challenge, the vaccine efficacy was really poor, down to 20%. So I don't fully understand why the vaccine efficacy is lower in endemic populations. These people will be semi-immune. These are adults. And it may be that there's some complex immune mechanisms at play here that counter the effects of the vaccine. Also, in the case of Mali, where you're exposing people to wild parasites, there's a lot of genetic diversity. And that, as we'll see going forward in this talk, is really a critical problem. Another vaccine that is targeting the liver stage is the Mosquirix vaccine produced by GlaxoSmithKline. This is the only malaria vaccine that has actually been licensed by the EU, and this is called RTSS. So as I said, this also targets that same sporozoite stage, but this time, instead of presenting the immune system with the entire parasite, what they've done is focused in on one particular protein called the circumsporozoite protein, or CSP, that is a very abundant protein expressed on the surface of the sporozoid. So what they've made is what's called a recombinant vaccine, or a subunit vaccine, where they make that CSP protein, or a part of it, synthetically in the lab, and they design it so that it includes a number of B and T cell epitopes that can stimulate an immune response. And really, for historical reasons that I don't fully understand, they conjugated this vaccine, or the CSP protein, to the surface antigen of hepatitis B. So this is the vaccine. It's delivered with an adjuvant to broadly stimulate the immune response. Now, there have been trials for RTSS over the last 10 years or so. And at first, the results were very encouraging. Here we're looking at the vaccine efficacy, looking at months after vaccination. And we can see that in the original trials, which are shown in green, we saw about 68% efficacy over 6 months. So this was encouraging. But then what you see is that that vaccine efficacy quickly drops. So now what they've been doing is looking at additional doses. So in blue, this is a 3-dose regimen of the vaccine, and you can see that when you get the vaccine efficacy continues to be poor. And now with 4 doses, which is the current regimen, you see again going out to about 4 years or so, the vaccine is only at 15% efficacy. So although this vaccine has been licensed and is now going through another pilot phase, I think it's a phase 4 phase, where they're rolling it out to hundreds of thousands of kids in specific countries in Africa, there's still, I guess, lukewarm excitement about this vaccine, and really looking for other alternatives. Now, one of the issues with that vaccine is the antibodies wane. And so the group in Oxford, led by Adrian Hill, have designed another version of this vaccine called R21. This is also based on CSP, but it's a larger chunk of the CSP protein. It doesn't have that hepatitis B surface antigen, and it's packaged into a virus-like particle that should be more immunogenic. And the idea is that it will better stimulate the antibody responses and also focus those antibodies specifically on epitopes within the CSP protein. So the reported vaccine efficacy over 12 months for R21 is 77%. So this is really encouraging. And some of you may have seen some of the press about this vaccine back in the spring. It's hailed as a potential breakthrough, a game changer. But personally, I think this last quote is the most important, which points out that it provides hope, but more work needs to be done. We've been down this track many times with malaria vaccines, and we really need to see longer term data to really see if this will prove to be a more efficacious vaccine. And I just want to point out that one of the big challenges with the liver stage vaccine, so both RTSS and the PFS-PZ, is that they're targeting this early stage. So this could be an advantage because it's a bottleneck, right? So if you can block the cycle here, you can prevent everything that comes after that. But what this also means is that if this vaccine is not 100%, doesn't have 100% efficacy, and even a single sporozoid makes it through, this will lead to a full-blown infection. So that in itself is a very difficult part of this life cycle to target. So what I want to talk about for the rest of this seminar is a different approach that's targeting the blood stage vaccine. So here we're looking at this stage. So this is the stage again where people become symptomatic and so on. So a different part of this life cycle. And I want to specifically talk about a vaccine that's designed to prevent malaria and pregnancy. I'm calling this the case of VAR2CSA because I think this is an interesting vaccine story that really highlights a number of the challenges in vaccine development that are true for other blood stage vaccines as well. So this vaccine is intended to prevent malaria infection in pregnancy. I mentioned earlier on that kids make up the bulk of the infections and deaths from malaria, but pregnant women are also a major risk group for poor outcomes of infection. We only have data for Africa where it says about over 12 million pregnancies are at risk. But if we also take into account the women living in other endemic areas of South America, South Asia, Southeast Asia, this number will be much, much greater. Now, most of the effects of malaria in pregnancy that have been studied have focused on infection with Plasmodium falciparum. And we know that this can lead to very poor outcomes, both for the mother and for the fetus. The mother can experience very severe anemia, and this can lead to maternal death. What happens in malaria pregnancy is the parasites will actually accumulate or sequester in the placenta. This causes what's called placental malaria. And this accumulation of the parasites can block the exchange of nutrients and gas like oxygen from the maternal circulation to the fetus. This can lead to stillbirth, babies that are born preterm, babies born small, so they have low birth weight, particularly under 2,500 grams, and also cause intrauterine growth restriction. And we also know that children born of mothers who had malaria in pregnancy can experience longer-term cognitive or learning deficits. So this is a cartoon kind of illustrating what we think happens in the placenta when a pregnant woman is infected with falciparum. So here's a cartoon showing the villi and the maternal and fetal circulation. We have a cartoon here showing our red blood cells infected with Plasmodium falciparum, and you can see that they're kind of accumulating on the surface of the syncytial trophoblast, and this can then block that exchange of gas and nutrients to the fetus. Now, we also know that, that there is a specific parasite protein that is really a critical mediator of the pathogenesis of placental malaria, and this protein is called VAR2CSA. So that's shown on this cartoon here. So we have our red blood cell, we have the parasite inside, but the parasite expresses this VAR2CSA protein, which it exports and presents on the surface of the red cell. So here we're showing one variant of the VAR2CSA, which is shown in orange. And this VAR2CSA protein has a high affinity for a receptor that's expressed on the syncytial trophoblast from the placenta. Now this receptor is called syndecan-1, and on this receptor you have these chondroitin sulfate A sugars that can bind directly with the VAR2CSA. So the parasite is using this attachment to the placenta as a way of avoiding immunity. So this is one of the classic immune evasion mechanisms. It's avoiding immune cells, it's avoiding being cleared by the spleen, basically by hunkering down in the placenta and hiding from the immune system. But at the same time, having these parasites down to the placenta leads to inflammation and these other pathogenic effects that I mentioned. But because VAR2CSA is known to play such a major role in the sequestration, it really is an ideal target for a vaccine to protect women from placental malaria. So the ideal would be to find a way to of a vaccine that will elicit a very strong immune response, that will generate antibodies that will recognize the VAR2-CSA protein that's expressed on the surface of the infected red blood cells, and that these antibodies would prevent that binding of the parasite to the chondroitin sulfate A on the placenta. Now, one of the challenges with VAR2-CSA as a vaccine, in terms of making a vaccine, is that it's a very large protein. So it's about a 350 kilodalton protein. This is shown just as a linear schematic here, but this extracellular part here is probably about 300 kilodaltons. So this makes it very difficult to make it as a recombinant protein, like I talked about the CSP protein, which is made synthetically. That's a much, much smaller chunk of the protein compared to making the entire VAR2-CSA. Now, VAR2-CSA is characterized by these different domains. They're called Duffy binding-like domains, or DBL domains. And typically this protein has 6 or sometimes 7 Duffy binding-like domains. Now, what those who are developing vaccines against VAR2-CSA, because they couldn't make the entire protein synthetically, instead they focused on 2 DBL domains, DBL1 and DBL2. And what they've done is made recombinant forms of just this portion of the protein. And the reason why they focused on this portion is that many studies in the lab have shown that this is really the business end of the VAR2-CSA. This is the part that is thought to adhere or bind to the CSA from the placenta. And they've shown in preclinical studies that antibodies, raised against these specific domains can block that adhesion of the infected red blood cell to chondroitin sulfate A in vitro. So there are 2 different vaccines that target VAR2-CSA that have gone through phase 1 clinical trials. One is called PAMVAC and one is called PRIMVAC. So these trials were completed just a couple of years ago, and the results from these studies were reported in early 2020 and late 2019. Both of these vaccine trials had very similar study designs. So they were phase 1A/B dose escalating trials. So the initial primary focus is on safety. So the initial group of vaccinees would receive a very small dose of the vaccine. Once they know that this is safe, they then can escalate to the next dose, and the next cohort or next group would get a higher dose of the vaccine. So the primary endpoints were safety and also immunogenicity. So they wanted to characterize the antibodies that were made by the people who were vaccinated and see how well do they recognize VAR2-CSA, and can they block that interaction between the parasite and the placental receptor. They also compared different adjuvants, which I won't talk about today. So people were given 3 doses of the vaccine. The cohorts in the 2 studies were very similar. Each one had 1 cohort from a European population, either France or Germany, and 1 cohort from a malaria endemic area or a malaria endemic country, either Burkina Faso or Benin, so both in West Africa. The only real difference was that the PRIMVAC vaccine only included women, whereas the PAMVAC vaccine trial included both males and females. Now, the results from both of these trials were also very similar. They found that the recombinant proteins were very immunogenic, so they stimulated a strong antibody response, but that antibody response was directed against alleles of VAR2-CSA that matched what was in the vaccine. So these are called homologous parasites, and I have a cartoon here to try to illustrate that. So, for example, if the vaccine people were vaccinated with the particular VAR2-CSA allele, this purple allele, they made very strong antibodies that could recognize parasites that also express that particular purple allele of VAR2-CSA, and in fact they could prevent the binding of those parasites to CSA. But unfortunately, when they tested those antibodies against other alleles of VAR2-CSA, so in this case an orange allele, those antibodies failed to recognize VAR2-CSA on the surface of the parasite, and therefore they also could not block the binding of the VAR2-CSA on the parasite to CSA. So what this means is that in these trials, they had strong homologous protection or immunity, but no heterologous immunity. So the reason why this is very important is that in the wild, if you were to immunize all these women and they become pregnant and they're now exposed to a whole lot of different forms of R2CSA expressed in naturally circulating parasites, these vaccines would not be able to protect these women because those antibodies can only recognize the exact same allele that was found in the vaccine itself. Now, this is highlighted by some genetic studies that have really pointed out just how polymorphic the VAR2CSA protein is. So in this paper that was published a couple of years ago, they isolated parasites from pregnant women from a number of different countries around the world, and they sequenced the VAR2CSA gene, so this entire extracellular region. And what they did here in these two panels in B and C is they're showing you where the genes are different in the different alleles from these different parasites. So either here they have lots of insertions or deletions across the gene, or in panel C, they're showing where the nucleotide sequence, and therefore the amino acid in the protein, differs between a parasite, for example, from Ghana and a parasite from Cambodia. Now, if we zoom in on this area that's covered by those two vaccines, the PrimVac and the PamVac vaccines, you can see that these areas in particular are very prone to insertions and deletions, and also have some areas where it's highly polymorphic. So it'll be very challenging to be able to improve these vaccines so that there's better representation of all this genetic variation in the vaccine. It may be that with this new mRNA technology, you could have mRNAs that represent many, many different versions of R2CSA. I think that's a very interesting approach to try to deal with this issue of polymorphisms. But in my lab, we're taking a completely different approach. And I would say it's probably a very heretical approach that we're trying to make a cross-species vaccine against VAR2CSA. Now, I don't have time to get into all of the backstory to this, but and I'm happy to answer any questions you may have, but this is something that we discovered really by chance from work we were doing in South America, in Colombia, which is in a particular area where pregnant women are exposed not just to falciparum and VAR2CSA, but also to another species of Plasmodium vivax. And what we discovered is that antibodies against one particular protein from Plasmodium vivax could cross-react and recognize the falciparum VAR2CSA. And we found that one of these proteins is called PVDBP, or Duffy binding protein. Now, PVDBP is not a homolog of VAR2CSA. There is no homolog of VAR2CSA in the vivax genome at all. And in fact, it's not even clear that vivax parasites sequester in pregnancy, that they even accumulate in the placenta. But instead, this protein is involved in invasion. So both blood stage antigens, but with very different functions and expressed at different times in the asexual cycle. So although these two proteins seem really unrelated and they don't share a lot of sequence identity, they do share some structural similarities in that the PVDBP protein has a DBL domain, similar in structure to the DBL domains from VAR2CSA. So why would this potentially be an interesting approach to pursue to make a vaccine against VAR2CSA? Well, our idea is that if we think about this from an evolutionary perspective, the fact that an antibody against the DBL domain of PVDBP can recognize the DBL domains of VAR2CSA suggests that there are conserved epitopes across these two proteins. And because they have such different functions, these epitopes may actually be conserved, at least structurally conserved, and therefore not under the same kind of selection pressure that would lead to all those polymorphisms that really challenge the current traditional vaccine approach for VAR2CSA. So to try to explore this as a vaccine strategy, we had a really useful tool at our disposal, and this was a monoclonal antibody made from mice that had been immunized with the DBL domain from that Vivax protein, PVDVP. And this antibody was shared with us by our collaborator, John Adams at the University of South Florida. And in our previous work in the lab, we showed that this monoclonal antibody, which is called 3D10, that it recognized the VAR2-CSA recombinant protein by ELISA and could actually stain the surface of red blood cells infected with falciparum strains that express VAR2-CSA. And not just one allele of VAR2-CSA, several different variants of VAR2-CSA. So the purple, the orange, and another color, for example. So this again pointed to the ability of this antibody to recognize something that's really conserved between these two species and also between these different alleles of VAR2-CSA. And importantly, we also showed that this monoclonal antibody could block the binding of those parasites to the chondroitin sulfate A using our in vitro assays. So what we want to do as a vaccine is to basically elicit similar kinds of antibodies in humans that would be like the one that is expressed by this monoclonal antibody. So as a first step to doing that, we mapped, and John's lab mapped this as well, we mapped the binding site of this antibody in the PVDBP protein, and we mapped it to subdomain 1 or SD1. So the next step was to try to take that domain of the SD1 and make it into a vaccine candidate. So to do this, we decided to make it as a peptide vaccine because these are only about 31 amino acids, so it's very small, can be made synthetically. We knew from some of our own data that the structure of the peptide was very important to try to elicit those kinds of antibodies that would be cross-reactive to VAR2-CSA. So we used some interesting chemistry with some scaffolds to try to constrain that peptide into a specific structure. We then conjugated that peptide to a carrier protein like KLH or BSA to ramp up the immune response. And then we put that into, we immunized animals, either rabbits or mice, along with an adjuvant to stimulate the immune system and hopefully have them make antibodies. So we took the serum from these animals and then we characterized them for recognition of VAR2-CSA and to see whether they could block binding in vitro. So the first thing we did was to measure the endpoint titers in the serum of these animals, so to see how good are the antibodies that they made. And in both animals, we found that they made very strong antibodies to the vaccine itself. So that's this SD1, the first bar in this graph here, and the first bar in this graph on the right. Now, when we look at the cross-reactive antibodies, so these are antibodies directed against VAR2-CSA, we see that in the mice, not all of the animals made these antibodies. We have 3 out of 5. And those that made the cross-reactive antibodies had a lower titer compared to the antibodies against the vaccine itself. In the rabbits, we found that all 3 rabbits made antibodies to VAR2-CSA, but again, the titers had dropped by about 3 logs. Now, when we took, because these antibodies in the rabbit were quite of a low concentration, we did an enrichment protocol to enrich for these antibodies that are specific to VAR2-CSA, and then we asked whether they would stain the surface of falciparum infected red blood cells. So these are just some examples of different red blood cells infected with parasites. You can see that with the DAPI. And then in red, you can see the antibody from those rabbits that's staining the surface of these red cells and staining really quite brightly, even causing agglutination or clumping of those infected red blood cells. So this tells us that these antibodies really can recognize the native VAR2-CSA protein. But unfortunately, when we tested those antibodies in our inhibition of binding assay, we found that they were unable to block that binding to CSA. So we have a number of ideas about why. First of all, this is a polyclonal response in these animals, and it may be that because they're making antibodies to different epitopes within the vaccine, we're not always getting antibodies directed at the epitopes that we want that could actually block the binding. So we need to think about a way to further focus the immune response in these animals to avoid those off-target effects and really hone in to those epitopes that will block that the parasite's adhesion to CSA. So the strategy that we've used is to do peptide scanning. Essentially, what this entails is we take the amino acid sequence of the entire VAR2-CSA protein, and we did this for 2 different alleles or variants of VAR2-CSA, so we can capture some of that genetic diversity. Then we had a company synthesize 20 amino acid peptides that scan the entire length of that VAR2-CSA protein with an overlap of either 18 or 19 amino acids. And these peptides are printed on a glass slide, which is shown here on the left. And then we scan those slides with all those peptides using our mouse monoclonal antibody, the 3D10, as well as that rabbit antibody that we generated. And what we're looking for are regions of the VAR2-CSA protein where particularly the mouse monoclonal antibody will bind to both, and it has very strong binding, because those could be really important epitopes for us to target with our vaccine. So this slide is just to give you a bird's eye view of what this kind of data look like. It's really, they generate an overwhelming amount of data, and I want to give a huge shout out to my colleague Bart Hazes and my 2 new postdocs in the lab, Bernard and Daniel, who've really been combing through this data for the last couple of months to try to really tease it apart and interpret what would be viable vaccine candidates for us going forward. And essentially what we're looking for are these little patches of red where the antibody binds very strongly and where it will bind to both alleles of the VAR2-CSA protein. And then what Daniel, Bernard, and Bart have done is to transpose those patches onto the structure of the VAR2-CSA protein. So fortunately, their 2 cryo-EM structures of VAR2-CSA were published last year and the year before. So although I'd shown you this linear cartoon previously, here you can actually see the 3D globular structure of this very large protein. And you can see it in complex with the chondroitin sulfate A here. Excuse me. So you can see that there's a channel where this sugars will actually fit into the protein, and that's what mediates the sequestration in the placenta. So next, we took those red patches from our peptide scanning data and then add those onto the structure and really try to see are there any patches that are very close to this binding site for the CSA. Because the idea is that if we can generate antibodies that would target these regions here, they might obstruct that channel and prevent the VAR2-CSA from being able to bind to the CSA. So just to give you an example of one of these areas that we're really quite excited about, here we're looking at peptides in this particular patch. We can see it interacts with the CSA. And then what Bernard and Daniel have done is to go through hundreds of sequences of VAR2-CSA in various databases to see the level of amino acid conservation across different alleles around the world. So far we've looked at lab isolates and about 400 or so field isolates from parasites in West Africa, either Benin or Malawi and Ghana, and Malawi is in Southern Africa, and also looking in Kenya. And we're trying to expand that to as many different isolate sequences as we can find. But what really stands out for us and what's very exciting is that there are particular signatures in these regions that seem to be very highly conserved, which is what we had predicted, right? We predicted that there would be something in these epitopes that would be conserved evolutionarily. So what we're doing now is making our short list of our new peptides based on this combination of the peptide scanning data and the structural information. We have about 10 to 15 vaccine candidates that hopefully we'll be ordering this week. Next steps will be then to immunize mice and rabbits with these vaccine candidates and characterize the immune responses and see if we're able to generate a more focused immune response and ideally antibodies that would block that adhesion of the parasites to the CSA using various assays. And then once we can narrow down to a couple of vaccine candidates, then we get it really much deeper into the immunology of it, look at how durable are these antibody responses, how broad are they, do they elicit memory responses that could give you a profound long-term immunity against placental malaria. So we're very excited about these new candidates and look forward to seeing what these data will bring. And with that, I'd like to acknowledge many of the people in my lab who've led this work, particularly Katie Mitran. This was the bulk of her PhD. She really pioneered this work on a cross-species vaccine for malaria. And now Daniel and Bernard are taking over that work in their postdocs. And as I mentioned, I'm really appreciative of Bart's contributions to this project. It's really enlightened me in structural biology, and I feel like this is really taking the vaccine into a very sophisticated area of vaccinology. I also want to acknowledge Lauren, an undergrad in our lab, who did a lot of this characterization of the rabbit and mouse serum, and Angie, who was an MSc student who also did a lot of work on this a couple of years ago. And I'd like to acknowledge our many collaborators around the world who made this possible and our sources of funding. And with that, I'd like to finish with this. Maybe this crazy idea we have will end up in the graveyard of great ideas, but at the moment, we're still optimistic that this could be an interesting strategy to make a malaria vaccine that's a little different from the traditional approaches that we've seen before. And with that, thank you very much. Happy to take any questions.